Now, after discussing the quaternary structure of proteins, let's look at protein folding. Before we proceed, check in the comment section. I have shared there a mnemonic on how to remember the notes on protein folding. So let's begin. Why should a protein fold? Where are proteins synthesized and why should it fold? So what are the examples of these foldings? That's what we are essentially going to discuss in this video. So we know that proteins are formed in the ribosomes as a linear sequence of amino acids. So for this linear sequence to fit into its native conformation, it must start folding. So what are the examples of these foldings? So we have domain formation. So for domain formation, the one that is first formed is the one that is dominated by the alpha helix, the beta pleated sheets, and the beta turns. Domain formation also is characterized by self-assemblance. So it is a self-assembling process that depends on the primary structure. Another point that you need to remember about that is that it involves intensive interaction among the side chains of amino acid residues on a polynucleotide. Then you also need to know that a domain is normally governed by thermodynamic principles such as that one of energy. The next one that you are going to look at is a molten globule. This is the second step after domain formation. It is dominated by the secondary structure, while the tertiary structure is highly disordered. Now for native conformation, it develops from molten globule state after several minor conformational rearrangements. Now let's look at oligomer formation. So oligomer is formed from native conformation. Now in the case of multimeric or oligomeric proteins, after attaining several specific conformation, protomers or subunits may fold into native-like structures, which may give rise to what we call oligomers. Let's go through our notes. Protein folding. Proteins are synthesized on a ribosome as a linear sequence of amino acid residues. This polypeptide must fold during and following synthesis to take up its native conformation. So steps in protein folding, we have domain formation. In this case, alpha helix, beta pleated sheets, and beta bend containing domains are formed in the initial step of folding of polynucleotide chains. It is a self-assembling process and it depends on the primary structure. It involves extensive interaction among amino acids residue side chains of polynucleotide chain. It is governed by thermodynamic principle, e.g. free energy. Then we've also talked about the molten globule. The next step after domain formation, secondary structure dominates while the tertiary structure is highly disordered. Native conformation develops from molten globule state after several minor conformational changes and rearrangements. Oligomer formation, in the case of multimeric or oligomeric proteins, after attaining specific conformation, protomers or subunits may assemble into native-like structures, initially after some realignments. It ultimately gives rise to final conformation of an oligomer.